welcome to this talk. So I'll try to do two different things today in this talk. The first thing is I'd like to show you that actually even with commercial instruments, you record data, and from that data you can extract much more information than you typically do, and that you can do very often with actually software you can get from the outside, and you can do this on any commercial instrument. You just have to sometimes run them a little bit differently, but you'll see that. The second thing I'd like to introduce to you today is imaging fluorescence correlation spectroscopy. And having your cake and eating it too refers to actually that you can take actually in a light sheet microscope not only structure, you can dynamics. You can record both, or you can do even more. It depends a little bit on what you want to do. And to motivate that, let me start and give you an example. So if you take a look at that picture, what you see here is a peptide which is labeled but binding to a lipid bilayer. And if you look at that, perhaps you see at the lower right corner, it's a little bit darker than the rest, and you see a little bit of structure, but that's about it. But once I start the recording, then you see the dynamics. And now you see that in the different parts of your bilayer, you have areas like in the lower right corner, for example, but as well a little bit higher in the middle here, where you see that particles or peptides just bind and either bleach or unbind, while on the other parts you see actually particles moving around. So this gives you already much more information than just having actually a simple picture with structure only. And that is actually what I try to drive. I mean, if you record with a light sheet microscope, you can do the structure and the dynamics at the same time, with almost more or less recording in the same way. What do you require? In principle, you require something like single molecule sensitivity, which is actually not a problem because more or less all the commercial microscopes nowadays have actually scientific CMOS cameras or EMCCD cameras that are already sensitive to single molecules. You want to have small observation volumes. Small observation volume means you have a high spatial resolution, and you can go as well to higher concentrations, because the smaller the observation volume is, the higher in concentration you can go and still see single molecules. Then you want to have fast acquisition, so you want to have high temporal resolution. And lastly, you want to be light efficient, because you want to observe fast, but as well over a long time. So the solution to that, I mean, you can imagine that already, it's of course single plane evaluation microscopy or light sheet microscopy, where you have an illumination objective that creates a light sheet. That light sheet is basically like a knife. It's cutting through your sample and illuminating one cross section in your sample. And then under 90 degrees, typically you have a detection objective with a high numerical aperture. This actually projects then your the fluorescence from your sample onto a camera. And that has multiple advantages. The first thing is that you have a small observation volume, because you can have very thin light sheets. You can create light sheets that are as thin as about on the order of 1 micrometer. And then in XY direction, of course, you're limited by the optical diffraction limit. So the volume you can get is about a femtoliter or so. So you get very high spatial resolution. The second thing is that you can do actually very fast acquisition, because the EMCCD or scientific CMOS cameras can easily record thousands of frames per second. That allows you to do actually parallel detection on all the pixels, and in all the pixels you see the dynamics, but you as well fast enough to see actually single molecules move. And then lastly, if you think about light sheet microscopy, you illuminate really only that part of the sample that you actually record as well. That means you do not bleach your sample as easily, not as phototoxic as some other microscopes, where if for every single scan you illuminate your whole sample, like for example confocal microscope. So here on the right side, you see actually how these light sheets look in our system. So on the top part, you see the light sheet from the top. You see actually the beam raised here in the middle. And the lower part, you see the light sheet from the side. You see that's it covers actually a relatively large area field of view, and this gives you ultimately relatively low phototoxicity, and you can observe your sample for a relatively long time. Okay, so if you can record now so fast, like what you can do now is you can take a look at single particles, and once they enter your light sheet, their signal is going to be imaged onto your detector. And on the right side, I give you actually two different examples of 2 pixels. And if a particle is seen, you see actually that the intensity goes up. If the particle leaves a pixel, the intensity goes down again. So from the width of these peaks, that will tell you already something about how fast particles move. And you can imagine as well that from the number of peaks, etc., you can basically get as well actually concentrations in your sample. So as an example here on the right side, these are correlation functions. These correlation functions allow you to statistically treat your signal, and they measure the average width of these peaks you see on the left side. And there's 2 examples. For example, EGFP, that's a green fluorescent protein in solution, in the rhodomine PE, which is a lipid in a supported lipid bilayers. And because lipid bilayers, membranes, are about 100 times as viscous as for example, the cytoplasm, you see actually a nice shift of the correlation function to the right. So the wider the correlation function is, the longer a particle takes to go through a pixel. That means the slower it moves. On the lower part, you see actually an example of 2 rhodomine PE labeled supported lipid bilayers done with actually 2 different concentrations. So one has double the concentration than the other, and you see that nicely in the amplitude that you get actually a different concentration. So the amplitude in FCS encodes the concentration. The width of the correlation function tells you something about the diffusion coefficient. And you can do this now and use this now. Okay. And get now from a measurement in your light sheet 3 things. The first is the projection. You can get actually the intensity image. That's the one you typically collect with your light sheet. The second and the third one are now a maps of the diffusion coefficient, so the width of the correlation function, and the concentration n map, that is the amplitude of the correlation function for every single pixel. So you see actually now maps of the diffusion coefficient over your whole sample and maps of the concentration over your whole sample. And that is a power really of imaging fluorescence correlation spectroscopy in the light sheet. You get now much more information because you can't, the diffusion coefficient and the concentration, of course, you can't directly get from the intensity projections. Okay, so simultaneous spatial temporal resolution. So how do we do that? I mean, what we do is we take fast acquisition, which means typically 1,000 frames per second. And we do this with small pixels, right? The pixels can be 50 to 100 nanometers or so in object space. Now, if you think about this, this is kind of a way you would typically not do either high temporal or high spatial resolution. Why? Because the small pixels means that you record relatively few photons if you go fast. So that does not give you enough photons typically for neither for FCS nor for good resolution images. So what we do afterwards, we bin. So you can sacrifice a little bit of spatial resolution. So we bin in space. That's what you see on the left side. Now we get for fast acquisition enough photons to measure the dynamics of molecules. On the right side, we bin in time. So we give up a little bit of our temporal resolution, but now we get actually at every single small pixel sufficient photons to get very good resolution. But it all comes from the same data. So that allows you then afterwards to do dynamics and structural resolution at the same time. Okay, let's start with a couple of examples. So the first example is because EGFP, it's actually the transcription factor morphed in Drosophila.